Hey, great. Good afternoon, everybody. I'm Mark DeSouza. I'm a biochemist by training and been on the business side of biotech over 20 years. I've oh, okay. So Alex Silver, who spoke before me, you still can't hear? Okay. Oh, that's good. I'm good. All right. Well, so let's start over. Good afternoon, everyone. I'm Mark DeSouza. I'm a biochemist by training, been on the business side of biotech 20 years. Been CEO of a number of venture-backed biotech companies in the rare genetic disease space. My first entrepreneurial venture was in 2010 when I founded a company to treat Alex Silver's son and other children affected by dystrophic epidermolysis bullosa, which is often referred to as butterfly children because their skin is so fragile. I've done a number of other companies in pediatric genetic diseases, in several cancers, especially cancers of the skin. And at two of those companies, one's a Bridge Bio company, one's funded by Venn Bio and F Prime Capital. I met Stuart Marcus, who's an oncologist, whose idea this was to treat cancer non-invasively with a new technology that we call sonodynamic therapy. And you saw the videos just before this, and that's news to me because I'm not really in touch with patients directly, and it's just brilliant to see the effect our therapy is having. So I'm going to give you a little perspective on the new way to treat cancer. We all know about previous attempts and current practice, which is surgery, radiation, chemotherapy, new approaches that have been new for 20 years with antibodies and targeted antibody therapies like antibody drug conjugates, immunotherapy, and more recently, radiopharmaceuticals. So these are all new discoveries that have attempted to change the paradigm of treating cancer. And I asked the question, how many discoveries does it take to change a paradigm? And the answer is really only one, but the paradigm has to want to change and be ready to change. And I think that's where we are today. You heard from previous speakers about AI and new devices and so on. What we have here is a new drug-device combination to treat cancer. SonALAsense is a pioneer of sonodynamic therapy. We were founded in 2019. We're headquartered across the Bay in Oakland. We've raised about 25 million in Series A and B preferred financings. And we've used those proceeds in a very lean and mean way to get into the clinic. We are currently in 2 phase 2 trials, one in DIPG, you heard the video from Lisa, and the other in recurrent glioblastoma, the brain tumor that sadly killed Beau Biden, John McCain, and Teddy Kennedy. So we're seeking $5 million to fill out a convertible note round. We've already raised 10. And that's really a bridge to our Series C financing, which is a large financing. The Series C includes $50 million from return on mission investors. You heard on the last panel, we've engaged Norm and Full Sky Partners to help us with this effort. And I must say, being at the JP Morgan conference, that's really received a lot of traction, where this return on mission funding will really provide leverage for traditional return on investment funding for us. It obviously helps that we've got early but promising clinical data with sonodynamic therapy in these 2 rare brain tumors. So what is sonodynamic therapy? It's a drug-device combination. The drug is a compound called aminolevulinic acid, or ALA. All of us here make 300 milligrams of ALA every day because our cells need it to make heme. And you need heme in your mitochondria, which you might remember from high school biology is the powerhouse of the cell. It's a powerhouse of the cell because it contains a lot of heme proteins, heme-containing proteins that are involved in the transport of oxygen. We pair this drug, which is given intravenously, with a device. We're currently focused on the device at the top of the slide, which is this helmet-shaped device that fits on a patient's head and delivers focused ultrasound. You heard that in the video from Dr. Koshman, as really a new way to activate a drug to treat brain tumors non-invasively. And we're currently in 2 phase 2 studies in recurrent glioblastoma and DIPG. We have built a second device ourselves that we will use subsequently to treat bladder cancer, pancreatic cancer, and other cancers outside the brain. But currently, our focus of our Series C is very much on clinical trials with the brain tumor device shown at the top. So I want to talk a little bit about the science. I promise I'll keep it science light. This is one of the few science slides I have here. This is truly a precision medicine. All cells that consume and need oxygen need to make heme. And the only way to make heme is through ALA, our drug. So normal cells convert ALA to heme just fine. But tumor cells take up so much more ALA because they're growing so quickly. They're greedy. They take up a lot of ALA, and they actually run out of iron. So they cannot make heme, and they accumulate the precursor to heme, which is called protoporphyrin 9 or PP9. And that's a very reactive molecule. If you shine a blue light on cells that accumulate PP9, they fluoresce pink. And that's why you see the pink color here. And this principle has been exploited by 2 companies that have an FDA-approved imaging agent. Oral ALA is approved to image brain cancers. You see that over here. This is a glioblastoma patient who drinks an ALA solution, waits about 6 hours for their tumors to accumulate the pink stuff, the protoporphyrin 9. And then the surgeon does a craniotomy, shines a blue light, and you can see the tumors over here glowing pink. And that's how the surgeon can cut out the tumor. Obviously, they can't get every single tumor cell. And that's really the advantage of our technology, is it's non-invasive, and you can go in and activate the pink stuff to blow up the tumor cell. So that's what we're doing, is we're taking a nutrient and we're converting it into a smart bomb that blows up the tumors without affecting normal cells. And this is not magic. We've actually got clinical proof of concept. I'll share some data with you. ALA is also approved in the bladder to image bladder carcinoma, and you can see this over here, where you can see the specificity, really, of the protoporphyrin 9 in the tumor that glows pink. That's tumor. Everything else is normal. This is truly a platform. These are all different tumors in the brain, outside the brain, that take up ALA, convert into protoporphyrin 9, and can be targeted with ultrasound to kill the tumors without affecting normal cells. As a small company, we're staying focused, starting in the brain, and once we have clinical proof of concept, we will move outside the brain. The mechanism is very well understood. How does ultrasound activate protoporphyrin 9 to kill the tumor cells? It does it by a process called lipoperoxidation. So this is ultrasound, this is protoporphyrin that's accumulated in the tumors. What happens is you get an energy transfer to molecular oxygen, and that creates reactive oxygen species that blow up the cell membrane, and the cell dies. When the mitochondrial membrane blows up, you get the induction of what's called apoptosis, or programmed cell death. So there are 2 independent ways where the tumor cells are killed, and there's no effect on normal cells. We've conducted a first-in-man clinical trial at the Ivy Brain Tumor Center in Phoenix, and we showed that this mechanism is indeed how brain tumor cells are killed. And that's a whole talk on its own. I won't go into that. I'll just show you one piece of animal data that's now been reproduced in 3 independent labs. So these are rats in which a brain tumor has been implanted. And you don't need to be an expert here to see the tumor over here in the white spot, that the tumor grows over 28 days in the control, meaning untreated rats. You treat them with ultrasound and they still grow. You treat them with ALA, they still grow. So the drug itself does nothing, the device itself does nothing, but together, sonodynamic therapy, ultrasound plus ALA, the tumor is a little stable and then it disappears by day 28. We treated at day 7, 14, and 21 over here, 3 treatments. If you take the brains of these animals out and stain for tumor, you see no tumor in the rats at 28 days. So this was a cure in the animals, okay? And there was 100% survival of these treated rats, all the controls died. So we're going to be publishing this shortly. This was work done with our collaborator in Toronto. So at the 30,000-foot level, what is sonodynamic therapy? It's really a non-invasive procedure. We're now treating patients once monthly. So they come in, they get treated, and so far in over 30 patients, we've seen no side effects, nothing. And the kids go back home, they go back to school the next day. So we can see this as a monthly treatment to keep your tumor at bay and have a good quality of life. Because there's no other way to make heme, it's very unlikely that tumors will become resistant to ALA. That's a big differentiator of sonodynamic therapy to everything else out there.
Yeah, Marty. Only in the brain or anywhere in the body?
The Marty asks, is that only in the brain or anywhere in the body? It's a common property of all cells, not just tumor cells. Normal cells also need ALA to make heme. It's just that tumor cells take up 50 times more ALA, and only tumor cells accumulate protoporphyrin IX, the pink stuff you saw in the photos. Normal cells don't. And that's the selectivity here, why the ultrasound only can kill tumor cells without affecting normal cells. So I'm going to show you some data in DIPG that you just heard about in the video. There's data on glioblastoma as well. I'm not going to go through that in the interest of time. So DIPG, as you saw in the videos, is a devastating tumor. It grows in the pons, which is this region over here in the brain, between your brain and your spinal cord. The pons is responsible for breathing and heart rate and balance and blood pressure. So pretty critical. So by definition, it's inoperable. Sadly, the standard of care has not changed since Neil Armstrong's daughter died of it in 1962, radiation. And the effects of radiation are so terrible that these kids are given steroids to counter the side effects of radiation. And then you have these horrible side effects of the steroids that you see here. We are treating kids in our DIPG clinical trial post-radiation, at least 4 weeks post-radiation. And what as you saw in the video, their quality of life in several of the patients is quite remarkable. We're in the phase 2 phase now of our clinical trial. We finished the dose escalation phase in 9 patients, and we're enrolling another 18 patients over the next year and a half to really look at the impact of sonodynamic therapy, safety, quality of life, efficacy, survival. FDA has told us overall survival is really the endpoint for an approval. We've opened up clinical trials at 3 sites here at UCSF, at Children's National in DC, and at the Nicklaus Children's Hospital in Miami. And we'll be opening up another 4 sites this year. We're already seeing signs of tumor regression and extension of survival. But as Lisa pointed out, so importantly, the quality of life is really what matters. If you compare this to other therapies, CAR T, for example, you have to be hospitalized for a few weeks, right? There's some wonderful results that have been published with CAR T therapy. And in these rare diseases, we need as many approaches as possible because not every approach will work for all children. But what really is important to us is quality of life. So we're treating patients monthly now. We learned a lot from our first patient, who was a lovely 6-year-old girl. We treated her first in August of last year. She was our first patient. And FDA told us, you need to go slow, only treat half the pons, wait 30 days, then treat the other half of the pons, wait another 30 days, then treat the next patient, and so on. We've now treated 9 patients in the trial, as Lisa said, post-radiation. You can see this is a large tumor shown in the crosshairs. We treated half the pons this side, and then 30 days later, the other side, the tumor grew a little bit and then started to shrink quite dramatically in what's called a partial response. This child, after only 1 treatment, actually had pretty remarkable quality of life. She went back to dancing. She loved ballet. Her parents told us that her double vision was improved and actually reversed. We've seen that actually in a number of patients. And she got to make memories and spend time with her family. Sadly, her disease progressed in another area that we did not treat. It went to the lining of her brain, and she passed away in January a year ago. But she really was a hero. Her family are heroes for volunteering for this new therapy. She was the first child to be treated with this new therapy. And we learned a lot from her. Since then, we've been treating patients monthly. Ellie Benem, who's sitting here in the front row, is our CMO and amended our protocol. A lot of oncology drugs are given in 28-day cycles. We don't really know how often we need to treat, but it makes sense with an aggressive tumor like this to treat monthly. This is a second patient in the trial, and you can see she's now 19 months. Median survival with radiation and chemotherapy is 9.5 months. and she's almost twice that today. You won't know that she has a brain tumor. And, may that long continue. The third patient and patients 3 to 9 at various stages, what's shown here is a swimmer plot that shows survival. So this dashed line down the middle is 9.5 months. That's published data from a clinical trial of radiation and chemotherapy in DIPG. And already, 4 of our patients are beyond that median survival. One is off trial here. This is a child who sadly passed away. And these other patients who were all treated recently, we hope will continue to survive and do well. And that's what time will tell. And then we need to enroll another 18 patients. And then we go to FDA. We've got orphan drug designation, we have fast track designation, and we're taking a regulatory strategy similar to the rare genetic diseases I've worked on before. Ellie's worked on pediatric cancers when he was at St. Jude and working on the bone marrow transplant unit. This is what you saw in the videos. I'm not going to belabor this. We're seeing the kids gained weight. They grew. One kid grew 3.5 inches during the trial, right? One child had a crooked smile. That's now normalized. This has also been seen with CAR T therapy. So we know that DIPG, if you can kill the tumor cell, you can gain function. I'm going to skip the glioblastoma section just to tell you that we are running this trial at luminary clinical sites, all the major academic medical centers, opening up a few more. This trial has more patients in it. It's different than DIPG. We don't expect we'll see a gain of function here, but can we slow down the tumor growth? Can we provide a better quality of life? That's the goal here. We're still in the dose escalation phase. We're starting to see some pretty amazing results. This is a compassionate use patient treated at the Mayo Clinic. And sort of the silvery white stuff here, this is the tumor. You can see this lady had a very large tumor, almost a quarter of her brain. And after just one treatment, it started to shrink. So we're starting to see some promising results there. So I'm going to conclude by just giving you a sense of corporate development strategy. We're working here in a very rare pediatric brain cancer. And a lot of VCs turn to us and say, how am I going to get my return, right? And I tell them all the same thing. This is proof of concept. We're educating the FDA on a new therapy. And we're going from this rare, ultra rare pediatric brain tumor to glioblastoma, which is much, much larger. And then with the other device I showed you, outside the brain into bladder, pancreatic, and other cancers. We also plan to go into other brain tumors like meningiomas, which are much larger markets than even glioblastoma. And we think we can do this rapidly with a really capital efficient operation. Right now, if enrollment keeps up this way, we should be on track for an approval in 2026. And that really sets the stage to broaden the pipeline from there. And that, of course, broadening the pipeline, cancer is a big indication, very large markets. So here's where we are. We're looking to close out our series, our convertible note round. We've raised 10 million already. We have another 5 left. And we're having discussions here. The return on mission investor commitment has been incredibly helpful in attracting return on investment capital. And this is something that VCs and private equity firms who've done diligence on us find quite attractive because the terms are far easier for them if a third of our financing is borne by a return on mission investor. So I'm going to stop here and just leave you with a summary here that we're doing something different here. We're the first company to develop sonodynamic therapy into patients having good quality of life. We're seeing tumor regression, extension, and survival. None of the side effects of radiation or chemotherapy. Nothing. We're on track for an accelerated approval. FDA has been supportive with fast track designation. We expect an approval in DIPG, the pediatric cancer, in 2026, and a year later in GBM. And then really as a platform, you remember that those figures with all the pink tumors lighting up. The plan is to go into bladder cancer and then into other cancers of the genitourinary and gastrointestinal tract. I didn't mention this earlier, but the brain tumor device we've licensed from an Israeli company called Incytech in Haifa. We have an exclusive global license to them for use in brain tumors. And they've been in the business over 20 years. They've raised over 500 million. A lot of investment has gone into this. I didn't mention the team. We've got a fantastic team comprised of experienced drug developers, device engineers, and physicists, people who've worked in the rare disease and ultra-rare disease space. Our patient advocate, April Freeman, is right here on the front row. We have a very patient-focused view to drug development. It's the approach that I think differentiates us. We really care about patient quality of life. And with the success in clinical development will come a financial success because these markets are very large. Thank you. I'll take any questions.
A couple questions. A question about.
Yes.
You mentioned meningiomas.
Yeah, the question was, have we done any clinical trials in meningiomas? I'm just repeating your question. No, we haven't done any clinical trials, but ALA is actually in a clinical trial to image meningiomas. So we know meningiomas take it up, and you can image it just like glioblastoma. So there's no reason you couldn't target it with ultrasound. So we can go anywhere. So we've treated bihemispheric tumors. We've treated tumors that are supratentorial and infratentorial. It's all in the software and how do you guide the ultrasound to the tumor. And that's what Insightec has done for 20 plus years, is figure out how do you deliver ultrasound precisely. So we can bathe the tumor and the surrounding area.
Can you show the device?
Yeah. So the image is a bit small, but you see it here. So this is an MR machine, and this device, this helmet, is placed on the patient's head. So the patient is lying on their back with their head in the helmet. They go into the device. You image the brain, you find the tumor, and then you use the software to steer the ultrasound beam to the tumor and surrounding area. So you can do that for a glioblastoma or meningioma or others.
So the device, so the ultrasound is in this helmet.
Yeah, is the ultrasound in the helmet is the question. There are 1,000 transducers behind that orange material you see there that deliver the ultrasound. Are there questions in the chat?
I don't see anything on the chat.
Okay.
So there's an injection?
That's right. The drug is injected. You wait about 6 hours. The tumors accumulate the protoporphyrin 9. Then you go into the MR, target the tumor with ultrasound. Tumor gets killed. The adults are awake during the procedure. They get off the table and go home. The children, because they wiggle too much on the MRI, are sedated. So they usually spend the night and go home the next day.
You worked on a bunch of individual treatments. Has anybody done more than once, maybe?
Yes. So we've had several patients. I didn't really point this out, but several patients have been treated multiple times monthly. And here's the...
Multiple months or multiple times in 1 month?
Once a month for multiple months. So these purple triangles here are a single treatment.
So that's 4 treatments.
Yeah. Yeah, so the long gap in between was the time it took for us to, amend the protocol with FDA.
6, 7, and 8, they're just recently.
They just started. Yeah, that's right. Yeah.
Okay, so only one has so far has died. Was the one you didn't expect.
The one we treated only once. Yeah, yeah, that's right.
And so what are you probably don't know what the pricing will be on this repeat?
No, we don't, but we'll price this, hopefully on par with or lower than other drugs in oncology. Right now this is a drug device combination, so we'll have to go in and work with CMS to get a CPT code and all that. That's to come.
It's got to be cheaper than radiation.
Yeah, I have no idea. So that's something to be figured out. Yeah. They're absolutely new policies. FDA has been very supportive to bring this forward. And we're using, right now we can't deny patients standard of care. So we are treating them post-radiation. But the vision here is to go pre-radiation. No. That'll be next. All right.
Fantastic. Well, that's fantastic information, and not only, I will also share the information with other people that I know. But this is great, and we'll see you at dinner tonight.
Yes, I will be there.
Thanks so
Thanks,
much,
Marty.
Marty.